A comparative study on the anti-hyperlipidemic and antibacterial potency of the shoot and flower extracts of Melastroma melabathricum lins. Long title, but the summary is that the study is about testing whether extracts from a plant called Melastroma melabathricum can be helpful in reducing high levels of fats in the blood, hyperlipidemia. The researchers gave these plant extracts to mice with high cholesterol for a few weeks and checked if it made a difference. Results showed that plant extracts, especially one called MSC, more on that later, helped lower the high fat levels in the mice's blood. Additionally, it seems to protect the mice's liver from damage. The study suggests that these plant extracts could potentially be used as a natural remedy for conditions with high blood fat levels. This is the table of contents. First, let's talk about the background and establish a few things. Why is high cholesterol harmful? Well, according to Mayo Clinic, your body needs cholesterol to build healthy cells, but high levels of cholesterol can increase your risk of heart disease. With high cholesterol, you can develop fatty deposits in your blood vessels. Eventually, these deposits grow, making it difficult for enough blood to flow through your arteries. Cholesterol is a fatty substance that can accumulate in your arteries. It's not entirely bad, though it has a reputation for being harmful. Some types of cholesterol can actually be good for your health if you consume them in the right amounts. There are two main types of cholesterol: high-density lipoprotein, HDL, and low-density lipoprotein, LDL. HDL is considered good because it transports cholesterol to your liver, where it can be removed from your bloodstream. On the other hand, LDL is considered bad as it takes cholesterol directly to your arteries, leading to plaque buildup, which can result in heart attacks and strokes. Triglycerides, another component of cholesterol, act as unused calories stored as fat in the blood. Consuming more calories than you burn can cause triglycerides to accumulate, increasing the risk of heart attacks. This is a visualization by Cleveland Clinic of the differences between bad and good cholesterol and its effects on the arteries. The study I will focus on LDL. The study has a definition of atherosclerosis, which is arteries thickening and stiffening condition manifested due to plaque formation by oxidized LDL of abundant and deranged lipid metabolism. What is hyperlipidemia? Well, according to the study, hyperlipidemia is a long-term problem caused by the body not processing carbohydrates, proteins, and fats correctly. It leads to high levels of fat molecules like total cholesterol, TC, triglyceride, TG, low-density lipoprotein, LDL, and very low-density lipoprotein, VLDL, in the blood. These will come up later in the study. The main reason of hyperlipidemia is an imbalance between the calories you consume and the calories you burn, often linked to lack of physical activity. Additionally, genetic factors contribute to the high blood cholesterol levels. Having high levels of lipids in the blood connected to serious health issues such as cardiovascular disease, type 2 diabetes, fatty liver disease, kidney problems, reproductive issues, and osteoarthritis. LDL stands for low-density lipoproteins, as mentioned previously, and is made up of fats and proteins and contributes to plaque buildup in the arteries. This extra buildup makes the arteries less flexible and leads to hardening of the arteries. When cholesterol on the artery walls undergoes oxidations, it can be dangerous. Oxidation happens naturally in the body, but if too much, oxidized cholesterol is produced, and it can be a problem. Melastroma melabathricum lin, also known as MM, is one of the 22 species found in the South Asian region, including Malaysia. According to the study, the small shrub known as Melastroma melabathricum lin have been used its parts like leaves and roots for a long time to treat various issues such as high blood pressure. Researchers wanted to see how effective extracts from MM are treated on high lipid levels in a mice model. The researchers also tested with antibacteria, however, it will not be discussed in my presentation. It's known that plants of the same type can be different in their effects based on where they grow. In essence, the study aims to explore whether extracts from the Melastroma melabathricum plant can be a natural remedy for high blood fat levels based on its traditional use and potential health. The study mentions the use of liver enzymes, especially AST, aspartate aminotransferase, ALT, alanine aminotransferase, and ALP, alkaline phosphatase, in the context of evaluating liver function and damage in experimental groups. AST is an enzyme found in various tissues including the liver and heart, and the study increased AST groups in Group F, treated with atorvastatin, more on that later, and Group B, hyperlipidemic control, suggests liver stress or damage. ALT is another enzyme primarily found in liver cells. It is released into the bloodstream, and monitoring ALT levels is a common way to assess liver health. The study reports significant reductions in ALT levels in groups C, D, and E treated with the plant extract, indicating potential protection against liver damage. ALP is an enzyme found in various tissues, including liver, bile duct, and bones. Elevated ALP levels can indicate liver or bone disorders. The study reports significant reduction in ALP levels in groups C, D, and E, suggesting protection against liver damage. More info about the groups will be discussed further on. The study employs these liver enzymes as biomarkers to assess impact of plant extracts on liver health, especially in the context of hyperlipidemia-induced conditions on mice. The goal is to evaluate anti-hyperlipidemic and liver protective properties of the plant extracts under investigation. Part 2. The study. In the study, researchers collected parts of plant like shoots and flowers and turned them into extracts using different liquids. These extracts were named MSE, extracted with ethanol, MFA, extracted with acetone, and MSW, extracted with water. The goal was to see if these extracts could have health benefits. To test this, the researchers used mice and divided them into groups, receiving different plant extracts and dosages. The treatment of this experiment was persistent for 28 days, where the three groups received different plant extracts with different concentrations, and the impacts of the doses had evaluated by quantifying the liver enzyme markers AST, ALT, and ALP as mentioned previously, and performing histopathology. Histopathology involves the diagnosis and study of diseases of tissues by examining under a microscope. The study looked at how these treatments affected mice's blood and organs. The results were analyzed using statistical methods to see if there were any significant differences. Now on to the results. All groups showed a significant increase in body mass compared to Group A, Group D, and E, which received different doses, showed remarkable anti-hyperlipidemic effectiveness compared to Group B. In Group E, the increase in body mass was slower. Liver enzyme levels, AST, ALT, and ALP, increased significantly in Group F, but they were significantly reduced in Groups C, D, and E, indicating that extracts have significant anti-liver damage properties. Hyperlipidemic mice, in Group B, had significantly elevated liver markers. Group C, D, and E treated with plant extracts showed significant reductions in liver enzyme expressions compared to Group B. Group F, treated with atorvastatin, standard medical cholesterol reducer, showed higher level enzymes in compared to Group B. Now on to the impact on lipid levels. Hyperlipidemia induced a significant increase in all lipid levels and decreased in HDL level, aka the good cholesterol. Group C, with MFA, acetone, showed a reduction in all lipid levels with an increase in HDL. Groups D, E, F, ethanol, and the standard exhibited significant reductions in all lipid levels with an increase of that good cholesterol. Okay, that was a lot of results and information, but here is a brief summary. In the study Melostroma melibathricum, ethanolic extracts were evaluated for their efforts on toxicity, liver protection, and heart in hyperlipidemic mice. Liver markers increased significantly in hyperlipidemic mice, but extracts of the plant demonstrated a dose-dependent reduction, outperforming atorvastatin, the standard medicine used by doctors. Heart histopathology showed improvements with extracts, especially MSE, ethanol, resembling normal structure. Results suggest the potential of the extracts in improving hyperlipidemia-related complications. It needs to be mentioned there were certain limitations that were in the study. The study lacks phytochemical screening and antioxidant property analysis, which would enhance its precision and understanding of the extract's impact on the liver, kidney, and heart in more detail. Part 3. The connections. For the biochemical aspect, The study explores the impact of Melostroma melabathricum plant extracts on liver markers, especially focusing on enzymes such as ALP, ALT, and AST, which are liver enzymes. Changes in liver enzyme levels are crucial biochemical indicators of liver health and function. The article discusses lipid profiling in hyperlipidemia-induced mice, addressing the levels of triglycerides Tg, total cholesterol Tc, very low-density lipoprotein VLDL, low-density lipoprotein LDL, and high-density lipoprotein HDL, the good cholesterol. Hyperlipidemia is a condition characterized by abnormal lipid levels in the blood, and the study assesses the potential of extracts to modulate these levels. The study evaluates the anti-hyperlipidemic potential of plant extracts by examining their impact on lipid molecules, indicating a potential role in modulating lipid metabolism. On to molecular genetics. The anti-hyperlipidemic potential of plant extracts implies a regulatory role in gene expression related to lipid metabolism. This suggests a molecular genetic mechanism which the extracts may modulate lipid levels. Individuals may exhibit genetic variability in the response to extracts, impacting how genes involved in lipid metabolism and tissue structure respond to treatment. The article touches on genetic and molecular aspects, highlighting how extracts influence gene expression related to lipid metabolism. This suggests a role in maintaining genetic and molecular homeostasis. Final connection: homeostasis. The study investigates the impact of plant extracts on lipid levels, emphasizing the role in restoring homeostasis. Lipid profile, including triglycerides, total cholesterol, LDL, and HDL, reflects the body's attempt to maintain lipid homeostasis. The anti-hyperlipidemic potential of plant extracts indicates their role in restoring lipid homeostasis in hyperlipidemia-induced mice. The extracts contribute to bringing lipid levels back to balanced state. The study explores the expression of liver markers and enzymes such as ALP, ALT, and AST. Changes in markers suggest disruptions in liver homeostasis, and the extracts demonstrate the potential to restore normal liver function. Final part, the conclusion. MSC, the one with the smaller dose with ethanol, showed significant anti-hyperlipidemic effects, while MSC ethanol with a bigger dose demonstrated appealing results by exhibiting liver-friendly properties, making it a promising alternative to atorvastatin during hyperlipidemia. MFA with acetone showed a mild association with liver but lacked significant anti-hyperlipidemic effects. The study suggests the potential of MSE in treating hyperlipidemic properties. The primary focus of their study was to compare anti-hyperlipidemic effects of MSC MFA extracts using two solvents. They conducted experiments on hyperlipidemia-induced mice with high cholesterol diet and carbimazole. The hyperlipidemic diet consisted of cholesterol and coconut oil. Carbimazole, an antagonist to thyroid peroxidase, was used to reduce T3 and T4 thyroid hormones, leading to an increase in blood total cholesterol levels. Hyperlipidemia was induced in all groups except the normal control by administering HCD and carbimazole. This study investigates the therapeutic potential of Melostroma melabathricum plant extracts in hyperlipidemia-induced mice. The findings reveal promising anti-hyperlipidemic effects as evidenced by significant reductions in triglycerides, total cholesterol, VLDL, and LDL. Coupled with an elevation in high-density lipoprotein HDL, the good cholesterol, the extracts demonstrate a capacity to restore lipid homeostasis and mitigate the adverse effects of hyperlipidemia on the liver and heart. Further research could delve into underlying molecular mechanisms and explore translational applications of extracts in addressing hyperlipidemia-related disorders. In summary, this study demonstrated the anti-hyperlipidemic efficacy of Melostroma melabathricum plant extracts. Here are the references that I used. Thank you for listening to my presentation. The study was conducted by these people and Department of Plant and Environmental Biotechnology, Faculty of Biotechnology and Genetic Engineering, Sylhet Agricultural University in Bangladesh.